Okay, let's just get right into it. Most students absolutely bleed easy marks on the haploid and diploid topic. But after this explainer, I promise you won't be one of them. Welcome. Today we're unpacking a single, seemingly tiny mistake that routinely torpedoes target grades. We're literally talking about severe grade boundary risks, all because of how answers are structured. So let's decode exactly what Cambridge examiners actually want to see and how you can bulletproof your marks.
So picture this. You're faced with a complex, multi-step biological pathway involving chromosome sets. What's your very first instinct? Let's take a pause here. Predict it. Where do you think most students completely derail before they even write their first sentence? Is it a bad calculation? Maybe a forgotten definition? Or is it something else entirely?
Well, here is the painful truth. Ordinary revision absolutely fails you here. You could know your textbook so well you see it in your sleep, but the classic trap? It's using the right terminology in the entirely wrong context. It happens all the time. Students memorize facts, but under the pressure of the exam clock, they just drop these generic rehearsed answers into highly specific scenarios. They totally fail to link their knowledge to the actual data provided, and boom, just like that, the marks vanish.
Right, let's break this down. When the syllabus tests you on haploid, or N, it is definitely not looking for a basic textbook regurgitation. It's testing your mastery of the fundamental principles behind a single set of chromosomes. You've got to prove that your baseline conceptual framework is rock solid. It's really about knowing exactly what that single set represents in the entire, broader biological picture.
And then, the syllabus brilliantly turns up the dial. Diploid, or 2N, requires you to exhibit a highly connected understanding of two sets of chromosomes. But here is the absolutely crucial part. It has to be transferable. Cambridge wants to see if you can take this core concept and apply it to some bizarre organism or weird experiment you have literally never seen before.
Which brings us to exactly why flashcard memorization breaks down. In a real exam, you aren't just getting a simple, "Hey, what is diploid?" question. No way. You are presented with complex, multi-step biological pathways. Your underlying knowledge is being tested right alongside your ability to handle information and execute experimental skills. If you just rely on memorization, you're going to completely freeze up when the pathway demands logical, structured reasoning.
So to actually secure those quick wins, you need what we call the method mark strategy. Remember, marks are super often awarded for your process, not just that final conclusion at the bottom of the page. Top-scoring candidates approach problem-solving in this specific topic using a really strict three-step sequence. Deconstruct, solve, and evaluate. First, they identify the given info, then they break the problem down into logical steps to solve it systematically, and finally, and this is key, they check for reasonableness and make justified recommendations.
Let's look closer at that systematic processing. Whenever you encounter a haploid or diploid question, you should follow a standardized sequence. First, identify the key elements. Then, apply the relevant biological principles. Next, execute the problem with incredibly clear working. Verify your results to make sure they make sense. And finally, present them in the proper format. Honestly, that is the exact blueprint for building an airtight top-band response.
All right, time for a quick interactive challenge. Imagine a Cambridge-style scenario that's testing a novel, never-before-seen diploid organism's cell cycle. How would you actually structure your answer? Think back to the framework. You move from the core concept to the evidence provided in the prompt to your scientific reasoning and finally wrap it up with the specific examiner wording. Formulate it in your head right now. What specific elements absolutely have to be in there?
Let's compare a flawed generic answer with a true top-band response. A generic answer might state facts about diploid cells with perfect accuracy, sure, but it scores incredibly low. Why? Because it completely ignores the experimental variables the examiner gave you. The top-band answer, on the other hand, takes those exact same facts and applies them directly to the unfamiliar context. It actively uses the data, it references the specific pathway, and it actually proves analytical depth. And, the crucial point here is summarized perfectly by the people who are actually grading your papers. This is pulled straight out of the Cambridge Principal Examiner report. The classic mistake is providing generic answers not specific to the question context. Always link every point to the given scenario. Seriously, if you write down everything you know about haploid cells, but you don't link it to the specific data given, you are just throwing away easy marks.
This all builds right into the top 3 examiner warnings. First up, using the wrong terminology for the wrong context. Second, rushing and forgetting to check units, which happens all the time when calculating statistics under pressure. And third, providing answers that entirely lack specific context. Essentially, if your answer could apply to any generic textbook question out there, it is simply not specific enough for a Cambridge A-level exam.
Now, the stakes escalate dramatically when we introduce experimental data. Data interpretation skills in this specific area are make or break. You're going to be expected to extract information from really complex tables and graphs showing chromosome numbers or DNA content. You've got to interpret the relationships between variables, calculate the relevant statistics with super clear working, and most importantly, draw evidence-based conclusions. Skip any of these steps, you're kissing the method marks goodbye.
To truly lock in that highest mark band, you have to level up to evaluative thinking. Top-scoring candidates do not just accept data blindly. They question assumptions. They consider entirely different perspectives when presented with an unfamiliar biological context. And above all else, they justify every single decision and recommendation with cold, hard evidence, pulled directly from the scenario. That is the exact difference between an average student and a top-tier candidate.
But I want to leave you with a kind of terrifying thought. What if you do everything we just talked about? I mean, what if your conceptual understanding of haploid and diploid is flawless, your data interpretation is absolutely perfect, and your contextual links are rock solid, but you still score a flat 0 on a major essay question? How is that even possible? Well, it happens every single year to thousands of genuinely smart students.
The hidden culprit? Command words, the specific verbs used in the prompt, words like explain, describe, or evaluate. Let's recap this, because it's vital. Mastering haploid and diploid concepts literally means nothing if you don't link them to a specific context, and it means even less if you ignore the command verb. If you describe a process when the examiner explicitly asked you to explain it, all your flawless biology knowledge won't save you from a 0. So next time you sit down to tackle a past paper, ask yourself, are you actually answering the question they asked, or just the one you hope to see?